bonds responsible for the protein structure. We saw that it can be divided into two, that is covalent and non-covalent bonds. In the previous presentation, we saw two covalent bonds, that is the peptide bond and the disulfide bond. Okay. Now the non-covalent bonds, these are mainly of the rest you can see here. The first one being the hydrogen bonds. Hydrogen bonds, it is formed between hydrogen and oxygen, nitrogen, or such kind of atoms. Hydrogen bonds are formed by sharing of hydrogen atoms between nitrogen and carbonyl oxygen. Okay, specifically in the case of what you call the protein formation, protein structure formation. Okay, each hydrogen bond is weak, but when they come collectively, it becomes very strong. And a large number of hydrogen bonds contribute to the protein structure. You can see here one kind, it is between the hydrogen on the hydroxyl group and the oxygen on the carbonyl group. Okay, one. Secondly, it can also be between the what you call, just like you saw it in this case, the carbonyl oxygen and the amine hydrogen. Okay, actually amide hydrogen. So that kind of a hydrogen bond is also possible. So these are the different kinds of hydrogen bonds. Okay, so hydrogen bonds individually they are weaker, but then when they come in a group, it actually provides stability and strength for the protein structure formed. Okay. The second kind of non-covalent bond, it is a hydrophobic interactions or hydrophobic bonds. Okay, so the hydrophobic interactions, it is the nonpolar side chain. The nonpolar side chains of amino acids, they tend to be closely associated with each other in proteins. So when they are folded, for example, here it is folded, right? So this is one like a nonpolar chain, nonpolar side chain of an amino acid, CH-CH3 twice, okay, and of another amino acid, okay, from another part of the polypeptide chain, right? So these two come together, and when they come together, they closely associate together in the hydrophobic interactions. Okay, so here you can see it is the hydrophobic interactions, which is actually a non-covalent interaction. Okay, fine. So it is valine on one side and another valine on this side. There, the hydrophobic side chains, they interact through hydrophobic interactions. Okay, so as such, these are not true bonds. The occurrence of hydrophobic forces is observed in aqueous environment, wherein molecules are forced to stay together. What happens to avoid interaction with the water molecules? These hydrophobic side chains, they tend to remain close together. So this is not a true bond, but it is just an interaction when it is an aqueous medium. Okay, to avoid interaction with the water molecule. Okay, now the other next one is the ionic bond, okay, or the electrostatic bonds, and these bonds are formed by interactions between the negatively charged groups. For example, the COO minus, the carboxyl group of acidic amino acids, with positively charged NH3 plus groups of basic amino acids. You can see here, right? So this is not the carboxyl attached to the alpha carbon. This is actually the additional carboxyl group of the acidic amino acids. Okay, and this is again the amino group of the basic amino acids. This was the amine group which is attached to the alpha carbon. Okay, so this is the additional amino group of basic amino acids, and this is the acidic group of the acidic amino acids. They come together during the ionic bonding or the electrostatic bonding. Okay, so such kind of interactions are again non-covalent bonds, bond formation. Okay, so we have these kinds of bonds. Apart from that, we can also find van der Waals forces, and these are non-covalent associations between electrically neutral molecules, and they are formed by the electrostatic interactions due to permanent dipoles or induced dipoles, it could be. Okay, so these are the different kinds of bonds that are responsible for the stabilizing the protein structure. We saw that there are two kinds of covalent bonds, the peptide bond and the disulfide bonds, right? And then the non-covalent bonds includes the ionic bond or the electrostatic bonds between the amino amino group of the basic amino acid with the carboxylic group of the acidic amino acid, right? and the hydrogen bonds between hydrogen and oxygen or nitrogen atoms. Okay, between the amino acids, between different amino acids. Okay, and we have the hydrophobic interactions, and this happens when the protein is in an aqueous medium. Okay, and what happens during that? The hydrophobic or non-polar side chains of different amino acids, they come together, aggregate to form, develop the hydrophobic interactions. Okay, so these are the bonds which can, which actually stabilize the protein structure.